Hello, I am Zachary Mead, and this is my presentation on olfactory transmucosal SARS-CoV-2 invasion as a port of the central nervous system entry in individuals with COVID-19. So a little introduction to this study. COVID-19, what is it? It's a severe acute respiratory syndrome is what it stands for. The SARS part of coronavirus 2, SARS-CoV-2, which results in the illness, which is COVID-19. And COVID-19 has been what's been affecting us as the World Health Organization has declared it a global pandemic. So I'll just summarize SARS-CoV-2 as COVID sometimes. So COVID is known to enter the human host primarily by binding to cellular receptors of angiotensin converting enzyme 2, or ACE2. So COVID has effects in the central nervous system, focusing on neurological symptoms of the large majority of the individuals who have gotten ill with COVID-19. So in this study, it tries to look at different things that the COVID virus has to do on the neurological system. So the study shows the presence of SARS-CoV-2 RNA protein in specific parts of the neuropharynx and the brain. Specifically, this causes, SARS-CoV-2 causes thrombotic ischemic infarctions of the central nervous system, present with evidence of SARS-CoV-2 neurotropism. So SARS-CoV-2 can enter the nervous system by crossing the neuromucosal interface of the olfactory mucosa, thus exploiting the close vicinity of olfactory mucosal endothelial and neural system cells, including those delegated to olfactory sensory nerve endings. It specifically penetrates neuroanatomical areas, including the primary respiratory and the cardiovascular control center, the medulla oblongata. This is mostly where it's been found within the brain, because it only affects certain cells with the SARS, with the angiotensin converting enzyme 2. So there's a little bit more about coronavirus just for the sake of the study. There's 7 types of coronavirus that affects human. 2 can persist in the central nervous system. So a lot of the studies based off autopsies, where 48% of all investigated cases showed COVID RNA in the brain. Animal studies are also part of the study, and they help show where SARS-CoV-2 is capable of entering the brain upon the expression in mice of the ACE2 enzyme. So a little bit about the methodology results. So the picture on your screen shows the presence of SARS-CoV-2. It shows the detection of decreased individuals with COVID-19 of anatomically distinct areas mapped in the neuropharyngeal as well as the central nervous system. So as you can see on the screen, the yellow represents the SARS-CoV-2, and this is kind of showing it in different regions of the brain. So we're just going to talk about the yellow for the sake of this presentation. As you can see, this C will show SARS-CoV-2 in areas of the medulla within the brain. And then G is another photo within the medulla, and K is an image of it in the neuropharyngeal area. So why is this important? So little is kind of known about SARS-CoV-2 at the moment, and this is what a lot of this study kind of focuses on. But intact COVID particles together with SARS-CoV-2 RNA in the olfactory mucosa were found in neuroanatomical areas receiving olfactory tract projections of the SARS-CoV-2. The neuroinvasion, including, it shows it can kind of invade neurons, including in areas of axonal transport. So this morphological detection of these particles in the axons was very difficult to do, and low numbers of particles were kind of shown in a lot of areas, which was kind of expected. So the whole point of the study was to visualize SARS-CoV-2 in the central nervous system on the cellular level, and kind of lead up to further studies to aggravate the fact that it's really shown in the olfactory bulb in relatively small regions of the central nervous system, and it affects a limited number of neurons, which is not surprising. because it's really specifically a respiratory illness, but it has shown to have effects on the brain. Detection of the SARS-CoV-2 RNA in some individuals of the central nervous system really have no direct detection to the olfactory mucosa. So there has been some shown in the cerebellum through the autopsy studies. Interestingly, SARS-CoV-2 in the study they found carry leukocytes across the blood-brain barrier, so it can affect the leukocyte, therefore crossing the blood-brain barrier, which is kind of how it's thought to invade the nervous system. So another possibility is the additional the axonal route, which they think is very unlikely, just due to the makeup of the virus. So kind of in conclusion of this study, it showed that SARS-CoV-2 can occur in neural mucosal interface and transmucosal entry via regions of the nervous structures. This is followed by transport along the olfactory tract, olfactory bulb into the central nervous system. And this explains a lot of the symptoms that people are experiencing due to COVID-19, including their alterations of smell and taste, which is very prominent one among many people affected. In the future, COVID is a very new field of study. There's a lot of studies focusing around cadaver research, and this study is one of those. It's interesting to see what the future studies will show and the effects of neurological effects. People who survive the disease, long-term effects are really widely unknown. Thank you for participating in my presentation on SARS-CoV-2 and how it affects the central nervous system. Here's my reference. Thanks for watching.